absent here in the cytoplasm. But here we'll get some organelles which are membraneless. They are the ribosomes. These are the ribosomes. So ribosomes are present, and the ribosomes are only 70S type. 70S type, having one bigger unit, that is 50S, and a smaller unit, the 30S. And these ribosomes, they may be freely present in the cytoplasm, or they may be present in the form of chains, which is known as polyribosome or polysome. Polysome or polyribosome, they are present. But they are prokaryotic ribosomes are only 70S in nature, having 50S and 30S. Then next, now let us discuss this genetic material. The genetic material is attached to the mesosome, and the genetic material is it is naked. This is the genetic material. It is known as nucleoid, or also it is known as genophore. Genophore. Now, what are the characters? Usually it is naked. Naked means it is not surrounded by any membrane like that of the eukaryotic cells. Then there is this is circular. Circular. As you see, it is circular. Then it is single. Single means only a single DNA is coiled to give a circular structure. And it is double-stranded. Double-stranded, because DNA is always double-stranded, it is also double-stranded. It is supercoiled. Supercoiled because it is very much coiled. And histone proteins are absent. Histones absent. Then you may ask, if histones are absent, how the negative charge of the DNA will be neutralized? No, there is another alternative, that is known as usually polyamines. Polyamines neutralize the negative charge of DNA. So this is the character of this the nucleus or genophore, that is the genetic material. Now, in addition to this genetic material, there is also sometimes in some prokaryotic cells there is again double-stranded circular DNA is there, which is extra chromosomal. This is known as plasmid. This is known as plasmid. So what is plasmid? Plasmid is extra chromosomal, double-stranded, circular, and self-replicating. Self-replicating means it doesn't depend upon the replication of this main DNA. It can replicate itself. That is why the plasmids in a prokaryotic cell, it usually varies from 15 to 100, because it can replicate itself. And this plasmid contains some of the genes, but they are non-vital genes. They are not, because these plasmids are not present in all prokaryotic cells. So whatever genes it contains, they are non-vital. So what are the different types of genes that are present? Now it contains the A+. If this gene is present, then it is fertile. Then it also contains that is the antibiotic resistance, that is a R factor. That is a antibiotic resistant gene that we discuss usually in case of the biotechnology. Then some of them also contain the call factor, that is a collisionogenic factor. So these are the different that is genes they are present, and usually these plasmids are used as vectors in biotechnology. That is one of the important functions. This plasmid is usually free in the cytoplasm, but if it will be attached with the main one, Suppose this will be attached with this one, then the name will change from plasmid to episome. Episome. That means if it is free, plasmid. If it is united, episome. Now if this episome will be again free, plasmid. Again united, episome. That means plasmid and episome are same thing, but which word will use, that depends upon whether it is free or united with. So that is about this plasmids. Then in addition to all these things, inside the cytoplasm, there are also certain inclusion bodies. And these inclusion bodies are mostly the reserve food materials and gas vacuoles. So here there will be certain inclusion bodies. And the inclusion bodies are usually, that is mostly they are the reserve food materials like phosphate granules, cyanophycin granules, glycogen granules. These are the different reserve food materials, and they are usually not bounded by any membrane. That means no membrane surrounds them. And beside these inclusion bodies, there are also sometimes you will find the gas vacuoles. But these gas vacuoles are usually present in case of aquatic ferns, and that is usually cyanobacteria, and also in case of that is a green and purple bacteria. Usually the gas vacuoles are present. Now after these things, there are another two things. What are these two things? One is Pili or Fimbri, and then we'll discuss another thing. So the next thing that we'll discuss is Pili, or it is also known as Fimbri. Sometimes the Pili and Fimbri, they are the words are used to interchangeably, but this Fimbri, this is also known as type 1 Pili, and this Pili is known as type 2 Pili. Now what is the difference? Now usually the type 1 Pili or Fimbri, these are usually small structures, they are bristle-like, they are numerous, and their main function is attachment to host as well as to substratum. While the type 2 Pili is also known as sex Pili, and usually these are large in number, these are elongated, tubular, and they are one or two in number, and their main function is sexual conjugation. That means they help in sexual conjugation. So if we will draw the diagram here, now you see here some small structures are coming like this. Now these structures you can call as Fimbri or type 1 Pili. This is, we can call it is Fimbri, or this is type 1 Pili, because they are small and bristle-like structures. But sometimes you will find certain the elongated tubular large ones like this, then this is known as type 2 Pili, or this is we can call it as a sex Pili. And this Pili or Fimbri, it is made of protein Pilin. And still in addition to this Pili and Fimbri, sometimes the bacteria, they may have flagella for locomotion. And there are some bacteria where the flagella may be completely absent, or in some cases they may be present. So the next thing that we'll discuss is flagella, and wherever this if this flagella is completely absent, it is known as atrichous. Atrichous means no flagella. It may be monotrichous. So mono means one. So monotrichous means single one. Suppose this a single flagella there. So monotrichous. Then it may be amphitrichous. So amphitrichous, in case of amphitrichous, that is one flagella on either side. The next is peritrichous. Peritrichous. In case of peritrichous, the flagella are present all around. Peritrichous. Then it may be cephalotrichous. Cephalotrichous. So in case of cephalotrichous, there is a bunch of flagella at one end, and the other one is lophotrichous. Lophotrichous. In case of lophotrichous, bunch of flagella on either pole. So there may not be any flagella. Atrichous, and depending upon one, two, or many, these are the different types of flagella. And flagella is always made up of a protein that is known as flagellin. It is made up of protein flagellin. So it is made up of protein flagellin, whereas in case of eukaryote it is made up of protein tubulin, and it has three parts. That is the basal body, then hook, and filament. That means it looks like a structure like this. That means this is the basal part, and this is the hook. This is the basal body, and this is filament. So this flagella, whenever it is present, it helps in locomotion. So this is the entire structure, the size, shape, and structure of the prokaryotic cell, including the different types of cytoplasmic materials, and there are some important structures like the mesosomes and their functions. So I hope now you have a very clear understanding about the prokaryotic cell and the functions of different parts. Thank you very much. We wish you a very bright future, and God bless you.